Hello everyone, I'm Rihanna Lily Smith from Technology Networks, and today I'm delighted to be joined by Dr. Wenning Qin, the Senior Vice President in Innovation at eGenesis, with over 25 years of experience in gene editing, to discuss their latest study published in Nature, which looks at the design and successful transplantation of kidney grafts from genetically modified pigs into non-human primates. This study could bring the technique a step closer to clinical testing. Firstly, why are pig organs promising donor animals for xenotransplantation?
Compared with a non-human primate, the porcine donor really stand out as having several key advantages. They are comparable to humans in organ sizes and function. And then on top of that, the gestational period is relatively short and litter size is large. So therefore, although non-human primate in terms of the genetics is more compatible with the humans, and because of all these key advantages, porcine donor really stand out as a more promising donor for human renal transplantation.
Can you talk about the challenges surrounding xenotransplantation using pigs and how these challenges inspired you to explore the use of gene editing to improve their compatibility?
In our porcine donor, we introduced 69 genomic edits. These edits can be grouped into three subsets. The first set involves knockout of three genes contributing to synthesis of glycan antigen on porcine cell surface. The second group of edits involve integration of 7 human transgenes into the porcine genome and expression of these genes in the porcine cell environment. The third set of edits involve inactivation of the porcine endogenous retrovirus element in the porcine genome, which mitigate the risk of zoonosis. So by introducing this collection of 69 genomic edits into the porcine genome, we have now engineered a porcine donor that is not only efficacious, supporting life long-term in non-human primate, but also should be safer because of the reduced risk of zoonosis.
What do you see as the most significant implications and ethical challenges associated with xenotransplantation?
This study really is a significant achievement for xenotransplantation research. First, it shows that the porcine donor can be extensively edited to improve compatibility with the humans, and then also to be safer because now the reduced risk of zoonosis. And then on top of that, an organ that had been significantly edited has been shown to survive and support life in non-human primate long term. The organ shortage is a global health crisis that continues to worsen. Porcine xenotransplantation is a promising approach, saving lives for patients suffering from end-stage organ failure. In eGenesis, and the welfare of the porcine donors and the safety of our patients will always be our number one priority.
Combined with immunosuppressive treatment, the transplant provided long-term primate survival of up to 758 days. Can you talk about this survival time frame and what this means for moving this technique closer to human testing?
In our study, we have several comparison groups, the 3KO alone, 3KO on top of that with the 7 human transgene expression, and then 3KO with the 7 human transgene expression, and in addition, carry retroviral inactivation. Comparison of the graft survival in non-human primate suggests that addition of the 7 human transgenes significantly improve graft survival. from a median survival of 24 days in 3KO kidney to 176 days with the 7 human transgene addition. So this suggests that human transgenes confer additional protection. On top of that, in vitro analysis suggests that introduction of the retroviral inactivation does not negatively impact graft survival, and in vivo data also support that these grafts also support life long term. We are very pleased with this proof of principle study in non-human primate, and will continue to work with the US FDA to advance renal xenotransplantation to clinical studies.
To make sure you never miss a story, subscribe to Breaking Science News, the daily newsletter from Technology Networks. Bye.